Okay. The second speaker for this session is Alexandria Volkening from Northwestern University.
Thanks, Mason and Heather. So I'm really excited to be talking with you guys today. I hope everyone is safe and well. So today I'm going to talk about how patterns form on zebrafish and how we measure those patterns. And this is joint work with Melissa McGurl and Bjorn Sonstedt. So they're both at Brown University now, but Melissa will be transitioning to Spotify next week. So my research broadly falls in the field of self-organization and collective behavior. So I'm interested in problems when you have individuals that are coming together to create some collective group dynamic that is bigger than any one agent. So the first thing you might think of is things like schooling fish or flocking birds, or in the olden days, we would think of pedestrian crowds, but this will be hopefully us at the next conference. But self-organization also occurs at smaller scales. And so this is where most of my work has focused. So I study this fish here. This is zebrafish, or Danio ririo. And what is really cool about zebrafish is that those patterns form because various cells, many different types of colors of cells, self-organize to create the patterns. And so that's what you're seeing in this movie here and at the timeline at the bottom of the page. And so I think it's remarkable that you have these tens of thousands of cells, and somehow they're able to reliably and kind of robustly create stripes. So you get dependable stripes on this fish, despite the fact that the cells are interacting in a noisy, inherently messy, biological, stochastic environment. So one of the big goals of my work is to better understand how do cells interact robustly and reliably so that we get nice, dependable stripes in this fish. And then the other big goal of my work has to do with mutations. So the cool thing about zebrafish is that they don't just have stripes. So if you get a genetic mutation on this fish, you end up with all sorts of mutant zebrafish. And so I'm showing you just a few of many different mutant zebrafish that can arise at the bottom of your page. So what you should notice, right, is there's zebrafish spots and lambert curves and kind of tortuous stripes. So from a mathematical perspective, this is a nice place to study pattern formation, just because there is this rich diversity and types of patterns that can appear on the fish skin. And they're all forming on a domain that's growing substantially. But from a biological perspective, the reason that we're interested in these patterns is that the biologists know for each of these fish what gene is associated with the altered pattern. So that's the word you see at the bottom of each picture. So for instance, Leo or leopard, when you have a mutant in that gene, you get a spotted zebrafish. And this is a gene that humans also have in common with zebrafish. So what that means is that from a biological perspective, there is a link between genes and the visible pattern we see on the fish skin, the phenotype. But what is not known is how the change in gene translates into a functional difference in how the cells are behaving. So what we want to do from a mathematical modeling perspective is put forward descriptions of how the cells behave normally, and then by changing cell interactions to create altered patterns, establish this yellow link so that we can link cell behavior to altered patterns. And if you can do that, you kind of think of all these links together, then what you're kind of doing by combining these biological and mathematical approaches is coming up with a way of linking genes to their functional impact on cell behavior and then how the fish pattern is looking. So this is kind of the pie in the sky goal of my work, is to link these various different scales in zebrafish and its relatives. So along this line, most of my work has focused on developing predictive agent-based models that can describe pattern formation on zebrafish and on mutant fish. And so to give you a sense of this, my models work on the same time and length scales as the biological data. So I'm going to show you some movies. But just to orientate you, these movies represent a third of the fish length, the body length, and the full fish height. Time is measured just like it would be measured in fish development. Every single cell that you'll see in these movies represents one cell on the fish skin. And we're going to simulate roughly from 3 weeks old to about 2 months old of fish development. So I'll show you wild type first. And so you can see the pattern forming kind of sequentially as the movie runs. To give you a sense of this, we're reproducing every quantitative measurement we can get our hands on here. These are things like the distances between pigment cells, the speed at which they move, and the time points in development when you should see new stripes emerge on the fish skin. So that's fine. And then the next thing you can do is take your model and try to reproduce mutant fish. So these are two mutant zebrafish that emerge because there's only some subset of pigment cells on the fish skin. So this guy doesn't have any orange or yellow cells, and this one has no black cells. So you can simulate this fish by just turning off that cell birth in your model, and this one too. So instead of talking about the model in detail today behind these simulations, what I want to do is talk about some of the questions that come out of these movies, because there are questions that come out of them. So when I show this movie here, the initial question that I usually get is, are the stripes straight enough? And the reality is, I don't know. There aren't quantitative measurements of stripe straightness over here, and so we don't know if they're straight enough. But I'd like to. And so what I'd like to do is to develop cell-based methods for measuring the curvature of these patterns. And then for these fish, this one and this one, I think there are much richer questions, because you may look at this pattern and this one here, and you may say, we may all agree, we think these look pretty similar, right? Eyeball norm-wise. But if you look at these two, I think depending on our collective mood over Zoom today, we may decide that this is a good match, or we may be like, I don't know. So the challenge is that with both of these fish, what I was doing when I was trying to reproduce these patterns was kind of by hand changing different cell interactions in the model and trying to reproduce a qualitative description in the biological literature of, in this case, messy blue spots, roughly aligned in stripes, in a silver background with peppered black cells. So you can ask, did we do that or not? And for this one, what we want is kind of orange globs in a blue region, and the orange glob should kind of sort of have a certain kind of width. And so what I'd like to be able to do instead is to quantitatively describe these fish patterns, understand what makes them different, and then you could automate matching your model to these patterns. And kind of to add to the complexity here, one of the other big challenges is that so wild type fish are pretty reliable, but mutant zebrafish are quite messy. So you're not necessarily trying to live or die by reproducing a pattern that looks just like this. In contrast, you want to reproduce realistic variability in your stochastic simulations, but you don't necessarily know what that variability should be, because in the literature, oftentimes you only see one or two characteristic examples of that pattern mutant shown. So you don't really know what the variability is. So what we'd like to do is kind of move past these qualitative comparisons and develop methods for quantitatively describing these fish and understanding the variability that emerges from the cell interactions. So along that line, this all boils down to an interest in developing interpretable, automated methods for quantifying cell-based patterns. And we want to do this in a way that specifically uses the agent-based nature of the data, just because we've put all this time into developing agent-based models. So today I'm going to talk about two things. My overarching goal is going to be to, like I said, to motivate questions related to quantifying these patterns. So I'm just going to give you a taste of our original model. My motivation there is just to, hopefully it'll remind you of some of your own models to give you a sense of the structure of it. And then I'll show you some of the sample predictions that come out of it, but I'm really going to sweep most of this under the rug. And then the other part of my talk will focus on raising questions relating to quantifying zebrafish patterns and going through our methods, which are methods based in topological data analysis, which Melissa McGurl is an expert in. So I'll mention here, if you have questions that we don't get to, you're welcome to email me. We can kind of simulate what a coffee break would be in a normal conference. I'll also say that I'm giving a longer talk specifically focused on the agent-based model at an MBI workshop on Friday that's virtual. So if you're trying to decide what to do with your Wednesday through Friday this week, you can drop into that conference. It should be very nice. Okay, so I'm just going to give you a taste of the biological background. So what you need to know about zebrafish is that there are 3 main types of pigment cells on the fish skin. Once you take into account various subtypes, you end up with 5 types of cellular agents or actors. I'll just call them black, silver, blue, orange, and yellow cells. And then these cells can interact in 4 main ways in the fish skin. The first is that they can just move. So you see this movie here of black and yellow cell repelling and attracting each other on the fish skin. They can also be born and they can compete with each other and kill each other off. And then the last thing they can do in very specific ways is change their type. So an orange cell can adopt instead a yellow type, and a silver cell can adopt a blue type. And so when you do that, you're kind of adopting a new set of rules of behavior. And so I think these kind of general types of interaction, of movement, changes in the number of agents in your model, and also changes in the types of agents in your model could be present in all sorts of kind of self-organization applications. And just in case you're interested in knowing more about the prior models that were present in zebrafish, I've highlighted the cellular automaton, so discrete space, agent-based, and continuum models from the last 2 decades. And so those are all models focused on 2 specific cell types on the fish skin. But in the last few years, there's been more data that's shown that all of these different cells are important. And so that is what kind of motivated us to develop our model. So to give you a broad overview, our model has those 5 different cellular agents that are interacting on the fish skin. It might start with about 100 agents, and at the end of a simulation, we have about 10,000. I don't take into account the cell shape in any way. So for me, m sub I is just the coordinate, the xy coordinate of the center of the ith black cell. X sub d sub I is the coordinate of the center of the ith orange cell, and so on. And so then these cells interact both locally and long range. So one of the neat things about zebrafish is that the cells can kind of locally interact with their neighbors through communication channels, but they also interact quite long range by reaching out these dynamic cellular extensions. So what you see over here is a cell kind of reaching out and pulling back these long extractions, and that's how they're communicating with their neighbors. And these legs could be up to half a stripe width in distance, so they're quite long. So as the domain grows, the cells move. That takes the form of coupled differential equations. I'm not going to show these, but it's just one ODE per cell describing how it moves. And then we also have cell birth, death, and shape changes or type changes. And those all take the form of stochastic discrete time rules for updating your domain. And so I wanted to mention here that where our model is seen as descriptive and where we're just kind of taking known biology and putting it in a mathematical modeling framework is in this area, so cell movement, birth, and death. And where we are trying to make predictions about the unknown signals behind cellular interactions are here. So what I'm going to do is just give you a taste of what these rules look like, just to kind of orientate us into the model. So specifically for cell birth, death, and shape transitions, we take into account those local interactions and the long-range interactions that the cells are doing. And the way we do this is suppose we are interested in this black cell at the center of the blue ball here. What we would do to determine its behavior is we'd look in its local neighborhood, that blue ball, and then we'd also look in a long-range annulus or donut surrounding the point. And so that annulus is representing the types of cells this cell could communicate with through its long extensions in kind of a homogeneous kind of way. And so just as an example of one of those rules, one thing we know on zebrafish is that black cells like to have more orange cells at long range than black cells. So the way we account for this would be to say, look at that center black cell, count all the black cells that are in a long range annulus neighborhood. If that number of black cells is greater than, by some factor, the number of orange cells in that annulus region, then with some probability, kill off the black cell in the center. So all of our rules have this general form of looking at a local neighborhood, looking at a long-range annulus around each point or cell, and then determining how that cell is going to behave based on the types of cells in those 2 neighborhoods. So for cell birth and death, we knew what types of cells should be in those two neighborhoods and how our rules should be structured. But for shape transitions, because that was really new biology, we did not know what these rules should look like. And just give you a sense of our approach to identifying these unknown cell interactions or the form of what these rules should be for instructing a cell to change its type. We broke the biology up into separate classes for modeling, evaluating, and predicting. So in particular, we decided that we needed to have one model that could reproduce these 6 fish. These are wild type and 5 mutations that form without any altered cell interactions. You just have cells missing. So you want consistency across your model with these 6 fish. Once you have consistency with those 6 fish, you step back, say, okay, that's our model. Let's see if we can reproduce something we didn't kind of build in. And so for that, we use all sorts of well-understood experiments yes, okay. So all sorts of well-understood experiments where we know how the biologists are changing the patterns. And then the final step is to make predictions about these altered cell interactions and mutations. So to give you a sense of some of our predictions, here I'm showing just a few of the patterns from that last class of patterns. And what I want to mention here is that when I was trying to reproduce these fish patterns, what I was doing was, like I said, by hand changing cell interactions in the model in an effort to reproduce a leopard spotted pattern or one of these other types of mutations. And I think the challenge with this is it's, like I said, an inherently subjective process. And if you look at some of these mutants, so this is a leopard mutant, this is a different gene called shockbret, and this is a gene called Lux. Once you start looking at those patterns for a while, they start to look pretty similar. And so how are we sure that the cell interaction we changed to create this pattern is not actually the cell interaction that should go with this spotted mutation? And so what we'd like to do, like I said, is to develop methods that can quantitatively describe these patterns and identify how they're different and identify the types of normal variability that we see in them. So if you look at zebrafish patterns for a while, what this ends up boiling down to is our wish list is that we would like to be able to, without looking at patterns, automatically identify the number of stripes that are present in an image. We want to be able to identify the number of spots, the stripe width. Like I said, we want to look at stripe curvature. We might also want to be able to get our hands on if the spots are aligned, and if the spots are circular or kind of elongated. And so our approach for this is based in topological data analysis and specifically persistent homology. Okay, so I realize I think that this is probably too broad of an overview for this audience, but I am fairly new to persistent homology. Melissa's the expert in this area on this project, so I'm going to just give a very broad overview of persistent homology from my perspective. But the idea is that you have some discrete data. So for instance, you might have these black, you could say they're your black pigment cells. And you might want to look at that discrete data, kind of eyeball it and say, there's kind of like 4 globs, and they're kind of sort of surrounding a region of empty space. And so in persistent homology, what you do is you put a ball around each point and you slowly grow the size of the ball. And when 2 balls connect, you draw a line between those 2 points. And so then there's nice computational resources out there for counting the number of connected components or globs in your data, and then also the number of holes in your data. So a hole right here, or a loop, as a function of that ball size. And then so what you're interested in is how does the number of connected components and the number of loops in your data change as a function of that ball size. And you want, in this application, we want numbers that remain for a large number of ball sizes. So what we're going to do is take this type of approach and reinterpret it as a way of measuring pattern formation on zebrafish. So in particular, what I'm going to show you is our methods applied to in silico, so agent-based modeling data at this point. But what we do is we take our model and we loop it to a periodic domain. So our fish is a cylinder in this case. And what we're going to do is apply persistent homology to the pigment cell coordinates. So you can see right here, as you increase that ball size, you pick up that, you can see that there are 3 connected components right here, right? So for us, because we're using this periodic domain, a connected component is really capturing a spot, and a stripe is a loop, is the presence of a loop in our data. And so that's how we get those 2 quantities. And then the next thing we want to get our hands on in an automated way is stripe width. And you can notice that as you kind of increase these ball sizes, where 2 of them connect, you can get an estimate on the stripe width based on twice that ball radius, right? So we see that when our loops start to die and merge, that's an estimate for our maximum stripe width. So once we have the number of stripes and spots in our data, we can cluster our cells into that number of stripes. So here we know that there are 3 stripes in this image. So we cluster our cells just using hierarchical clustering into 3 stripes. Then you can trace out the boundaries of each cluster and measure how straight those boundaries are relative to a perfectly straight stripe. So that's our measurement of stripe curvature. For spots, same kind of idea. Once you know the number of spots from persistent homology, you cluster your cells into that many spots. You can identify the centers of your cells and do principal component analysis to look at how varied they are, whether they're kind of round or elongated. So that's our measurement of spot roundness. So what I'm going to do now is just show you these kind of measurements applied to some in silico data. So this is those measurements applied to 1,000 simulations of our wild type model. And here we're breaking up the patterns based on what fraction of different patterns we see in the simulations. And to give you a sense of this, in the past, what was going on was that I was kind of by hand viewing 100 patterns and doing this classification by eye. So this allows us to handle much more data. Then the other thing you can do is look at these patterns that up to now have only been described qualitatively, where we see orange globs or messy blue spots. And so what we can do by looking at thousands of our simulated images is start to identify what we think is characteristic for these patterns, what spot size should be characteristic as predicted from our model, what type of variability we expect to see as characteristic from our model. And so if you define these distributions as characteristic, then you can start to push your model further, add in more noise, and ask, when do you fall outside of these distributions? So that's what we're going to do here. So remember, I said that the way the cells interact in our model is that they have some donut around them, and they interact with all the cells in their donut. That's not particularly realistic, right? What we really want to have is cells kind of extracting and extending cellular extensions in real time. But as a first step to this, what we're going to do is instead of specifying a set donut radius, we're going to change that donut radius randomly for every single cellular interaction, every single time step. And what you want to do is just build more and more noise into the cell interactions and ask, when does your model break down? So what I'm showing is, as a function of adding more and more noise into our cell interactions, what fraction of wild type looks normal? And in comparison, what fraction of this mutation look normal as you add more noise? And so what we see here is that particularly there's a lot more variability in the mutant patterns than in the wild type patterns. That's what we'd expect. The other type of thing that comes out of this analysis is that we're able to predict what types of variability we expect the biologist to see. So here, what we expect is that when these patterns break down, they should see patterns that are only smaller than characteristic spots and less defined. So once we have large classes of in vivo data, we can test these predictions. And so that's what we're doing now. I've been working with the Nusslein-Volhard lab at the Max Institute for Developmental Biology to extract cell coordinates from detailed images And we're also looking at applying functional persistence to kind of lower quality, lower precision images as well. So that's what you guys will see at the next conference in person. So I'm going to stop there. In conclusion, what I've been working on is agent-based models of pattern formation on zebrafish and its evolutionary relatives. And what we've particularly been focused on in this talk is asking questions about measuring pattern variability and comparing in vivo data to in silico data for zebrafish. So I've put the references here, and I want to acknowledge my collaborators again, Melissa McGurl and Bjorn Samsted. Thank you guys. I hope you're all well.
Thank you very much. If anybody has questions, if you send me a private ping, and then I will ask you to ask your question. You don't have to write down the question to ping, just ping me that you have a question.
Mason, there was one that came to me during the talk.
Okay,
So from Liz Bradley. I'm gonna go ahead and unmute you so you can ask your question.
Hello, now that I've got my camera working, that was a great talk. Thank you so much. That first movie you showed, I was just curious, I know that's not your work, but I was really curious about how one makes a movie of a fish that moves and grows and have it kind of stationary and watch the spots form.
That's an excellent question. That is one of 2 movies that I have seen in the zebrafish biology literature in the last 20 years, I'd say. So I'm not sure how that's from Shigeru Kondo's lab in Osaka University. I'm not sure how they did it, but I know that they must be hard to do because there's not a lot.
Okay, there's another question that's come. This is from Yanis Lazovskis. So please unmute yourself and ask your question.
Great, thank you for your talk. I was wondering about the model. You talked about these rules that the cells have. And I was wondering if the movement and the transition of cells in the future, because this happens over time, if that's impacted by the cell movement and transition in the past, sort of like if they have a limited, I don't know, amount of energy, or maybe they can only transition a limited number of times.
That's an interesting question. I've never thought of it in terms of energy before. What we have looked at is whether or not there's kind of a signaling delay in when you interact with your cells and when you actually behave. So we've built that kind of thing into our model. At this point, we're not really tracking cell behavior, individual cell behavior in time. It would be really interesting to look at what the trajectory of a single cell's life is in the simulation and to compare that to the data. So that's a good question, but I haven't seen any bio on that specifically about the energy.
Okay,
Great.
we have.
Thank you.
Oh, sorry. We have one final question, and I'm going to ask for them since they don't have their microphones set up. And the question is from Peter Bork, and the question is, what are your thoughts on investigating the structure of tumor growth with these tools?
So Bjorn and Melissa and I are really interested in looking for more applications, particularly for, we think, undergraduate research, to kind of take these tools and apply them to other data sets, both in silico and in vivo. We'd love to try other data sets. We just haven't yet.
Okay, so with that, let's thank Alexandria again. Let me stop recording briefly here.